Welcome to the American Physiological Society's Living History Project. This project was initiated in 2005 by the Society in order to develop an audio-visual record. The record is in the form of interviews with those senior members of the Society who have made significant contributions to the advancement of physiology as a science and as a profession. I am Charles Wonder. At this time, it is my pleasure to interview my colleague, Dr. G. Edgar Folk. We are both emeritus professors of physiology at the University of Iowa's Department of Molecular Physiology and Biophysics. Ed, I understand that from a very early age, you have been influenced toward physiology. Could you tell us who were the first people to exert such an influence?
Charles, there's no doubt in my mind of the first influences. It was my family. My father was a Methodist minister of the British curate type of naturalist, and we lived on the edge of a large woods in our small town of Andover, Massachusetts, and our recreation was walking in those woods because we did not have an automobile. In fact, our family didn't have an automobile till I was 21 years old. At any rate, we walked in the woods, and my two parents were very tuned into nature, and my father would turn over logs and identify the salamanders under the logs, and there were many questions posed to, there were 3 children in the family, and the questions were posed. For example, there's a songbird there that lays 1 egg each season. And there's another one over there that lays 4 eggs. Why do you suppose there's such a difference between those 2 birds? That's the kind of question I was brought up with. In addition to that, my father had another background. He was a very ardent and very successful gardener, and of course raised the usual fruits and vegetables. But he was especially fond of raising flowers and was very good at that, and used to compete with having the most beautiful flowers. But then he went beyond that. He decided that he wanted to produce a new kind of gladiolus, and so he started cross-pollination and controlled pollination. And so he developed that so far that he gave talks on that. Therefore, the conversation around the dinner table was the experiment. So I was brought up with that word. I think that shows a very early influence.
Well, I think there are other people who influenced you, some even before you entered college. Could you tell us something about these other people who influenced you toward the life sciences?
We were very fortunate in that town because there was a preparatory school called Phillips Andover there. And they had a very high scholastic rating and high standards of education, and they were able to help their students into the most high standard colleges. And the biology teacher there was a big influence upon me. Larry Shields was his name, and he encouraged people to think in terms of experimental biology. He liked to talk about the experiments. And so he was an obvious influence. Furthermore, he gave me a position as a manager of a large room where they kept specimens of plants and animals. Another major influence was another student called Donald Griffin, and we were close friends, and even eventually had a bird banding station together. And this was an important influence because I, at that point, got the concept of the concept of quantitative work in the field, outdoors. And I should tell you about Donald Griffin because he became a very well-known scientist, and we were friends all through college as well. Don became a member of the National Academy of Science, and one of the discoveries that he was well known for was the echolocation found in bats. In fact, he invented the word echolocation. And I had the fun in graduate school of actually witnessing and going to his experiments on echolocation. At any rate, Donald Griffin and I he was a very stimulating person to me the rest of my life.
You attended Harvard University from 1933 until 1947. You received your A.B. there in 1937. Then you received your Ph.D. also from Harvard in 1947. But let's focus now on your undergraduate years. You've already mentioned that Donald Griffin continued to exert some influence on you when you were an undergraduate at Harvard. Who were some of the other people that did have quite an influence in directing you toward physiology when you were an undergraduate at Harvard?
Well, this may surprise you. I'm going to start with a person that I worked for in the summer. He was a contractor for large buildings named Clark Maycomber. Clark Maycomber was on the Board of Regents of Harvard University, but his family was known as the skiing Maycombers because his son George was on the Olympic ski team. At any rate, while I was working with them, they gave me a present of a complete ski equipment for downhill skiing. And I became addicted to that. Therefore, with other people, we went skiing in the mountains in New Hampshire. However, that's an expensive sport in that some people stay at places, ski resorts, but we did not do that with my friends because we joined the Harvard Mountaineering Hut and a club, and they had a hut near the top of Mount Washington. So we did our skiing on Mount Washington, living at the Mountaineering Hut, which is near the top. Now, I'm mentioning this because later on, I was known as an expert on cold weather physiology, and I got my first experience of that walking through deep snow to the Harvard Mountaineering Hut. It was a very good way to spend a Easter vacation, and we were right near a exciting place to ski called Tuckerman's Ravine, which is a bowl a mile across. So quite often, two of us would enter Tuckerman's Ravine and be the first to ski there in the morning, but later on in the day, there would be as many as 1,000 people skiing in Tuckerman's Ravine. At any rate, I was used to cold weather camping, and I had to make use of this during World War II. Now, the other important person at that time was Donald Griffin, and he interested me in the physiology of hibernation, and he had a grant, even as an undergraduate, to ban all hibernating bats in New England. And so he formed a team, and we were all on the team, and we actually did ban 16,000 hibernating bats. Now, this, of course, he wrote papers about their migration, but I think one of the most important things that came out of our work was the age of bats. Remember, many of them are the size of the white mouse that you keep in the laboratory. Now, small animals don't live very long, usually. The white mouse usually only lives 2 years, or at the most 3 years. But some of our banded bats lived long enough to give us a record of 30 and 35 years, so that this was an important finding that came out of our work. But there was one other major influence at that time. That was a postdoc fellow at the biology department. His name was Herman Rahn, and Herman Rahn became later the president of the American Physiological Society. Herman Rahn was a very large influence on me at the time. His experiments had to do with the hormones of reptiles, and he'll come up later in my story because I collected reptiles for him and kept his experiments going. So Herman, by the way, he became a well-known experimenter because he wrote a book later on the diving women of Korea, and also he was well known in high-altitude physiology. There's a famous plot in the high-altitude area called the Rahn-Otis plot. So this gives you an idea of what an influence this early person was upon me.
Well, now let's move on to your graduate career. Tell us something about the role that your graduate advisor played in stimulating you toward physiology.
The graduate advisor became much more than that. Actually, when I was at Phillips Andover, I came in to visit Harvard University because I had a cousin who was a graduate student. His name was Sears Crowell, and he became a professor at the University of Indiana. At any rate, Sears introduced me to a professor called John H. Welch. And I was very impressed because he took the trouble to sit down on some stairs and talk to me for half an hour. And we discussed the appropriate courses to take at Andover in order to be prepared for biology at Harvard. So this was, Jack Welch became my advisor in graduate school. Actually, another influence, even as an undergraduate, was Jack Welch's advisor. There was a professor called G.H. Parker, who was one of the first to study the nerve impulse. And actually, he was the lecturer in my first course in biology at Harvard University. Now, he had been Jack Welch's advisor, and Jack Welch became my graduate advisor.
With respect again to your graduate career, were there any interruptions in your graduate career?
Well, I was there for 2 years and fulfilled the coursework for the PhD. And I also wrote 2 papers on hibernation while I was there under the encouragement of Jack Welch. Those were papers in a refereed journal, and they influenced the rest of my life, I think, because I enjoyed assembling data together and then writing a technical paper about it. So that phase and getting the coursework done was completed. But in the middle of that, in the middle of the 2 years, Jack Welch introduced a new idea to me. He had done experiments at a biological station in Cuba, the Atkins Institute, and he had also done experiments at a biological station in Bermuda. And he thought this was part of training. So he arranged for a fellowship for me to go to the Atkins Institute in Cuba, which is located in a rainforest as part of my training in biology. So I designed an experiment which was to study the effect on the reproduction of bats, tropical bats, the effect of latitude and climate on their annual reproduction. And was fortunate enough to complete that experiment. And this was because when I left there, the laboratory sent specimens to me so that we could study the animals the whole year round. This was a very exciting experience and exciting part of training. At the same time, Herman Rahn came into the act again because he had discovered in reptiles that there is the corpora allata in the ovary of some reptiles. And he wanted me to collect broadly through Cuba and Haiti, to collect reptiles and preserve the ovaries so he could look for them in a variety of reptiles. Well, then the Smithsonian Institute came in the act, and they put in some money so I could go to Haiti to fulfill what Herman Rahn wanted. And I did go to a very remote part of Haiti where few scientists had been and made these collections and came back at the end of the summer and then took on my second year of graduate work.
Were there any other interruptions after that in your graduate?
After this 2-year period, I left graduate school for 2 reasons. There was the possibility of being drafted, and also I wanted to be a conscientious objector in a war that might come up. So I decided that was not practical and that I should become a non-combatant in the military. So I joined the National Guard and I eventually became a sergeant in the First Aid Corps, in the Medical Corps of the National Guard. But at that time, I had run out of money to pay tuition, and so I was teaching at St. Mark's School in Southborough, Massachusetts. At the end of 3 years there, I was about to be drafted, or the National Guard would be called out. So I had a friend who knew about the Merchant Marine, and since this was non-combatant, I left St. Mark's with a close friend from the faculty and went and joined the Merchant Marine and was actually assigned to a ship. My friend was Hale Very, the music teacher at St. Mark's School, and we were both assigned to ships at the same time, and Hale Very got on his ship and went off. But I got a telephone call just before my ship left, and they said, We have a military laboratory at Harvard University called the Fatigue Laboratory, and we understand you have experience in winter camping, and we want you to come to run a environmental chamber at minus 50 degrees and work with some soldiers we have here as subjects. This is a military laboratory, so please come. And so I left the Merchant Marine and spent 4 years at the laboratory called the Harvard Fatigue Laboratory.
Your going to the laboratory, was that a further interruption of your graduate studies, or did it somehow help you conclude your graduate studies?
Well, to jump ahead, while I worked at the Fatigue Laboratory as on staff for 4 years, as they had done before, they allowed me to complete my thesis and get a degree from the Department of Biology. This had happened previously to a famous professor of exercise physiology, Sid Robinson, and to Steve Horvath, who became what could be called an environmental physiologist.
Harvard Fatigue Laboratory, back at that time, it was certainly a famous institution, and it's still well known by people working in environmental and exercise areas. I believe it closed in 1947, and unfortunately for others of us who are physiologists in other areas of physiology, we might not have heard about it or certainly don't know too much about it. Could you tell us a little bit about what we should know about that laboratory?
Well, I'll start with an illustration by a famous man. And this is a book written by L.J. Henderson. L.J. Henderson was an excellent biochemist and is known because students have to memorize the Henderson-Hasselbalch equation when they study biochemistry. L.J. Henderson was much more than a biochemist. He was a very deep thinker and a philosopher as well. But he founded the Fatigue Lab with the concept of the total environment of each individual, the homeostasis of the individual in terms of physiology, but also of their whole environment. In other words, L.J. Henderson wanted to go deeply into physiology, but to include the social influences upon man as well. This reputation spread of Henderson as a leading thinker and physiologist, and so many visitors came to be in his laboratory. And when I described to you what facilities were there in the laboratory, you'll understand why other scientists flocked to him. Now, I'll describe the equipment that L.J. Henderson put into that laboratory. In the first place, he wanted to study the person during exercise, and he wanted to study their blood and respiration. So he went deeply into two methods of physiology called the Haldane gas analyzer and the Van Slyke blood analyzer. And he actually lined up in the Fatigue Lab 5 of each of these instruments, showing how deeply he wanted to go into that. There was a treadmill with all equipment around it for measuring metabolism, and very famous athletes came and they were studied on that treadmill. Furthermore, L.J. Henderson and the later workers developed a desert room, a hot room with a treadmill in it, and a cold room that ran at minus 40 degrees. These were very large, so you could have groups of people in them and study, even exercise in the heat and exercise in the cold. So you can see why the laboratory became pretty well known to visit and to spend a sabbatical at. And it is true, people came from Europe to spend sabbaticals there. There was technical help, and it was a wonderful place to work. So this gives you the background. I had known of L.J. Henderson because I had read this book, and later on his position was taken over by a famous person called Bruce Dill, but I think I can mention more of him later.
Let's conclude our discussion of the Harvard Fatigue Laboratory with you telling us about the person or people who had the most influence upon you in developing yourself as an environmental physiologist.
First, I would like to mention that the history of the fatigue lab is carefully, before World War II, is carefully covered by Steve Horvath and his lovely wife, who was the daughter of Bruce Dill. This book is a well-known volume describing the early fatigue laboratory. The major person that dominated being at the laboratory was Bruce Dill, a professor at Harvard University. The first 2 years when I was there at the laboratory, he was an officer in Washington, D.C. He had a fine career helping the war effort and received the Legion of Merit for that. And Bruce Dill had studied at Stanford University and received an honorary degree from there. And then he was early in the development of the fatigue laboratory, and then its last 2 years he was the director. Bruce Dill published 300 papers, technical papers, from the fatigue lab during his career. You can imagine rubbing shoulders with a person like that. He was an extremely stimulating, friendly person who continued being my friend the rest of his life. Now, another person there must be described that you rub elbows with, and this was R.H. Rowton. Now, R.H. Rowton is very well known in physiology because with Meldrum, he discovered carbonic anhydrase, which allows us to breathe and allows our circulation to do its job. Now, part of the time, Rowton, whose primary appointment was in Cambridge, England, but part of the time at the fatigue laboratory, he worked on discovering carbonic anhydrase. And he made use of the 5 von Schleich apparatuses that were lined up there. Now here is a picture of Bruce Dill using the Van Schleich apparatus which Rowton used to discover carbonic anhydrase. I think that is enough about that person. As far as discussing other people, I should explain how the fatigue laboratory was set up. there was a person in charge of the hot room, the desert room, and that was Robert Johnson. And Robert Johnson also was in charge of developing developing nutrition supplies for the military. And for the cold room, Woody Belding was in charge, and there were many important projects under the leadership of these two people. I can mention the kind of things that were done in the laboratory. Of course, testing in the cold and the heat. But one of the most interesting projects was developing electrically heated suits for the bombers that were flying to Germany at 40,000 feet, and their cockpits were very cold. And so in the laboratory, we developed electrically heated suits with the heat placed appropriately at various parts of the body. These were the B-17 bombers that were responsible essentially for winning the war because of their bombing of Germany. Well, this was typical of the contribution of the fatigue laboratory. I would like to say that it was a laboratory with that reached out broadly. I was put in uniform twice. Once I was sent to Newfoundland, a very important assignment because I was with the Boy Scout named Seipel, who was with Byrd at the South Pole. He had now become a major in the Army. And also Henry Bassett, a famous physiologist who had always studied people when exposed to the outdoor environment. So we were on a team to teach winter camping and to take part in winter camping on the mountains in Newfoundland. Also, I was assigned on submarine patrol to help out the crew of submarine patrol. And that's why I had to be in uniform, because of being in hazardous duty. I think that's enough for the fatigue lab at the moment.
Okay. Well, after you left Harvard and its fatigue laboratory, you accepted a position in the biology department at Bowdoin College in Maine. I think you remained there for about 6 years. And I assume your primary assignment was teaching, but I think you managed to do some rather significant research at the time. Could you tell us about that research?
It was very interesting to be at Bowdoin College because part of the job was to be director of a biological station in the middle of the Bay of Fundy. And the Kent Island the Bowdoin Kent Island Biological Station. And so I went there summers with students, and we did research. I did, the only material you could work on were the birds of this remote place. There were eider ducks and many kinds of gulls, but the petrels came in from the deep ocean to dig burrows. And the significant finding I did learn was that petrels can hibernate, and I published 2 papers about the low body temperatures, normal body temperatures which are attained by petrels. At the same time, I had received, due to Woody Belding, a contract with the Army to study some human experiments, and this was the effect of impermeable material against the skin, a military question. And I could do experiments with the faculty and with students. So for 2 years before I left Bowdoin, I had this contract, and the person who made it possible, who worked with me, was Robert Peary Jr. He was the son of the person who had first reached the North Pole, and he lived in Brunswick, and it's a long story, but he was very pleased to work with me for 2 years, and the two of us published a monograph on the effect of impermeable material on the skin. That covers the Bowdoin period, I think.
Well, as we cover the Bowdoin period, we come now to the University of Iowa. And in 1953, you accepted a teaching position in the physiology department here within the medical school at what was then called the State University of Iowa. Why did you leave Bowdoin?
Well, it's a good question because I was very content there and enjoyed the life. But it was a 9-month appointment each year, and I had to try to find some way of earning money during the summer. And this was inconvenient. That was one reason, because Iowa offered me a 12-month appointment. But especially I wanted to work with Steve Horvath, who had preceded me at the fatigue laboratory, in the same position that I held at the fatigue laboratory, and he had a fine environmental chamber at the University of Iowa, and I was very pleased to join that department.
Could you tell us a little bit about your teaching responsibilities when you first came here, particularly with respect to the medical students?
Dr. Hines, Harry Hines, had very strong ideas about teaching in a physiology department. He wanted every faculty member to be associated with the medical course, and so we lectured in that course. Although we all had other courses to teach, I had a course of 144, which I taught for 25 years, which was it was called Intermediate Physiology for graduate students of other departments and for the dental students. At the same time, we lectured in the medical course, and we had conferences for each week with the medical students of maybe 20 in a conference to discuss the lectures. More importantly, Dr. Hines wanted us to do research with the medical students. So he divided the entire medical class into groups of 8. And each of us was to take 3 groups of 8 and do research. Actually, the system worked very well. The medical students liked it, and I actually published 3 papers with medical students as results of their hard work, which went on for a full long semester. More importantly than that, Dr. Hines asked us each to give an advanced course in physiology for graduate students. Well, I had always wanted to give a course entitled Environmental Physiology. And I was a little cautious about the term, but I had talked to a famous person at the fatigue laboratory who was on the staff, Per Scholander. And he was a very exciting person. We went and tested sleeping bags in the mountains. I went with him to sleep in the mountains for the military. And at any rate, at the end, as the fatigue lab was phasing out, and the fatigue lab phased out because it was the model so that they could build other laboratories around the country similar to the fatigue lab. And that was going on, so the fatigue lab was phasing out. I talked to Scholander about giving a course because I knew I was going to be teaching. And he said he was founding a laboratory at Point Barrow, Alaska, with Larry Irving of Swarthmore. And but after that, he wanted to obtain a boat and put equipment on it and go to the tropics to do experiments. Well, the laboratory at Point Barrow, the Naval Arctic Research Laboratory, became very famous. At any rate, I said to Scholander, now, when you do that sort of research, what are you going to call it? What kind of physiology? He said, I shall always call it expeditionary physiology. And I said to him, well, it's hard for a person in the physiology department to try to get a professorship in expeditionary physiology. So I've got a term I'd like to press, and that's environmental physiology. And we discussed that. He didn't like it very well. So that was the end of that, except I finally got my chance with Dr. Hines to give a course in environmental physiology. Well, after 6 years of giving that course, Lee and Febiger came to me and said, have you got anything to write a textbook about? I said I could write one on environmental physiology. So they said, okay, we'll do it. And so the first textbook of environmental physiology was then written. Here is the second edition here of the textbook of environmental physiology. And the reason that I am showing this is we tried to persuade the artist to depict Bruce Dill on the cover of this second edition. Now back to the first edition of physiology, environmental physiology. It was very interesting. I published that first edition in 1966, and I think it's the first time the term ever came out in print. 2 years later, there were literally 8 books with the title of Environmental Physiology. Now, this means only one thing. There was a perfectly good area of physiology, an area of study, an area of interest that lacked a name, and they finally had found a name.
Would you please discuss circadian rhythm -- biological clock, this topic in hibernating animals?
I actually have emphasized 2 fields which overlap. And one of them is mammalian hibernation, as begun with Donald Griffin. And the other was, my thesis was on circadian rhythms or biological clocks. So that these 2 overlapping fields occurred throughout my entire career. One is an influence on the other. Now, you asked a question about circadian rhythms, and this is under the control of sunrise and sunset. So in the laboratory, you can manipulate sunrise and sunset and give an animal a 4-hour light period or a 12-hour light period or even continuous light. And the biological clock in the animal responds differently to these different light periods. So this is called an experiment in photoperiodicity. Later on, after I had done quite a few experiments in the laboratory, I was invited to go to a place of extreme photoperiod, which is the Arctic. And the laboratory where Scholander and Irving, where they had founded a place to work, is at Point Barrow, and there in the summertime there's 82 days of the sun going in a circle overhead. This is an extreme photo period. Therefore, at a period of time, I was allowed to go up there to study the effect upon the animals and the Eskimos there of this extreme photo period. So that is an explanation of going from the laboratory into the field. At the same time, I had an interest in hibernation, and this is influenced by photoperiod. Photoperiod prepares the animal for hibernation. And later on, at that laboratory, I had a remarkable opportunity. Back to hibernation. I attended many meetings over the years giving papers on hibernation, and a frequent topic of conversation was, do bears hibernate? And everybody agreed that until there is a implantable physiological radio for use in difficult animals, we'll never learn whether bears hibernate. Fortunately for my program, a talented person wanted to get a PhD with me who had a background in electronics. His name was Warren Esler. And I explained the problem to him, and he said, I think we can do it. And Warren Esler, who, by the way, later became the head of all science at the University of Vermont for the rest of his career. Anyway, Warren Esler devised the Iowa transmitter for implanting into difficult animals. This is the Iowa transmitter. Now, this particular transmitter had been in the body cavity of a 1,000-pound polar bear. Now, by having this instrument and modifications of this instrument, we were able to study very difficult animals and find out whether they hibernate. The laboratory, which I've already mentioned to you, which was established by Scholander, is this laboratory here, this very large building, the Naval Arctic Research Laboratory. And this is what happened. I had been there studying the biological clocks of local small animals. I mentioned to the director that there was no information about the hibernation of bears, but that we now had an instrument in which we could determine whether they do hibernate or not. And Max Brewer was his name, and he said, we'll take that project on. I'll build cages for you, and we can study the hibernation of 3 kinds of bears here. So that launched that program of hibernation of bears. I must tell you more about that. We're talking about the situation of an animal in a den with a in that position, and how do you study its physiology? The radio capsule could also be used on other fierce animals. This is a wolverine. And wolves, we wanted to study wolves in order to understand their biological clock. Now, how do you persuade a very difficult bear to receive a radio transmitter. Well, fortunately, the zoological park people had developed a impact dart in which you can put a tranquilizer or immobilizer, which is a better word. Our transmitter made a record like this. It showed the heart rate, the EKG, and also recorded body temperature. So that these were the measurements with which we started work on 3 kinds of bears, black bears, grizzly bears, and polar bears, to determine whether they hibernate. Here is a picture of putting in a transmitter in a grizzly bear. Now, this was a remarkable instrument because it was long life. It did not have a distance capacity, but it did last for as long as 2 years, and therefore we were able to get long-term experiments.
You've talked about your biological rhythm studies with whole animals. Did you do any work with isolated tissue?
I had 2 programs when we began the study on the bears. There was one laboratory, and they were 4,000 miles apart, but it's remarkable how well the 2 laboratories worked together. I had long ago founded the Laboratory of Environmental Physiology at the university, and then there was the Arctic Laboratory. Fortunately, I had a efficient manager to run both laboratories, 2 managers to run the 2 laboratories, and we were able to get quite a bit of work done of a different type. It was necessary to continue the Arctic Laboratory for this reason. We were the first to show that all 3 species of bears do not eat, drink, urinate, or defecate for as long as 7 months. This, of course, had to be studied. And so one of our major works at the Arctic Laboratory was collecting the first sample of urine from the bears after they hibernated. And then we did chemical analysis of that urine sample. So that's a whole story in itself. But in the meantime, I was running a active a laboratory for bench work back at Iowa. And I had quite a few visitors come to the laboratory. I had postdocs and also had visiting professors from Europe. And we did quite a bit of bench work. I'll give you one example. In our study of biological clocks, it was considered that the clock within the body, all the organs of the body, are regulated by 2 organs in the brain, the pineal gland and the subesophageal portion of the hypothalamus. But the term that was used quite often was regulate. And so I asked a question about that. I wonder if the organs themselves have a pretty good clock and don't necessarily need to be regulated by the pineal gland. Therefore, we started a program of isolating organs to see whether they had a biological clock. For example, we isolated adrenal glands, and we found that there was a crisp 24-hour rhythm of the energy metabolism and the secretion of adrenal glands. And then we tried the same sort of thing with the heart, and we isolated perfused hearts with the Langendorf technique and kept them going for 2 days, and there was a very crisp biological rhythm in the beat of those isolated hearts. Furthermore, we studied heart cells and found a circadian rhythm in isolated cells. Therefore, perhaps the brain control should be called a guiding, not a regulator, but a synchronizer might be a better word because of the results of our experiments.
From the perspective of an individual who has achieved eminence as an environmental physiologist, there's certain advice that we'd like to receive from you. And one is, what kind of advice would you give to a beginning physiologist?
I've often talked to undergraduates about going into experimental physiology, and my advice is simplified by saying, I would say to them, if you can't help go into this field, then you should go into it. If you ask a question about whether you would like it or not, I suggest you try something else.
Okay, now, as the American Physiological Society is entering or has entered the 21st century, what kind of advice would you give to the Society's council and to its long-range planning committee?
I'm a great admirer of this society and the way it's been handled through its history, especially the last 50 years. And I think that they have helped the whole field of physiology and are interested in training young physiologists. They're doing an excellent job in making it possible for young physiologists. And I know that they are giving attention to making good physiology teachers. I think they should go beyond that. I think that those people in charge of graduate students are neglecting one other thing, and that's the presentation of their material. I've attended a great many seminars on physiology, and they're very badly given, and there's no training to, I think the professors themselves need training. For example, simple matters such as when a question is asked at the end of a seminar, please repeat the question. This should be automatic and a reflex as far as young trained physiologists are when they have to be chairperson for another person giving a seminar. And the seminars should be given to the person in the back of the room, not to the people in the front row.
As for physiology itself, what do you see in its future?
It's a very interesting period. I am already seeing experiments and papers being delivered with the combination of organismic, cytoplasmic work and genetics being combined with other kinds of organismic work. And this combination and fusion is the future, and it's already beginning. It's so pleasing to see.
Well, Dr. Folk, on behalf of the American Physiological Society, I want to thank you for this most interesting and informative session. It will be maintained in the archives of the American Physiological Society at its headquarters in Bethesda, Maryland.